Good day, everyone. Welcome to another session of our discussion entitled about automation. Automation is considered as one of the most important breakthroughs in the recent history of laboratory diagnostics. In a model of total laboratory automation or TLA, many analyzers performing different types of tests on different sample matrices are physically integrated as modular systems or physically connected by assembly lines. The opportunity to integrate multiple diagnostic specialties to one single track seems effective to improve efficiency, organization, standardization, quality, and safety of laboratory testing, whilst also providing a significant return of investment on the long term and enabling staff requalification. For the objectives, we have the following. So hopefully at the end of this discussion, each one of you will be able to achieve these. For the definition of terms, please become acquainted with the words presented on the screen. So I do hope that you will really familiarize and memorize the terms because these words will be very useful on our following discussions. Now let us start with the question, what is automation? The word automation is inspired by word automatic. So automatic means exercising control without interference. So automation means getting work done by machines which can run on their own without our continuous monitoring. Automation refers to machines with intelligence and adaptability, which reduces our workload and need for nonstop supervision. One benefit of automation is a reduction in the variability of results and errors of analysis through elimination of tasks that are repetitive and monotonous for most individuals. Now let's proceed to the history of automation. The era of automation arrived with the introduction of the auto-analyzer using continuous flow analysis and the robot chemists that automated the traditional manual analytical steps. Successive generations of standalone analyzers increased analytical speed, offered the ability to test high volumes of patient specimens, and provided large assay menus. A dichotomy developed with a group of analyzers devoted to performing routine clinical chemistry tests and another group dedicated to performing immunoassays using a variety of methodologies. Development of integrated systems greatly improved the analytical phase of clinical laboratory testing, and further automation was developed for pre-analytical procedures such as sample identification, sorting, and centrifugation, and post-analytical procedures such as specimen storage and archiving. So all phases of testing were ultimately combined in total laboratory automation, through which all modules involved are physically linked by some kind of track system, so moving samples through the process from beginning to end. The first true standalone instrument, not machines that are equated with manual laboring, was recorded in the laboratory in 1957. So the clinical chemistry autoanalyzer one used continuous flow analysis. By 1969, the SMA or sequential multiple analyzer debuted, and next came the SMA with computer by 1974. But before that, we also have the automatic clinical analyzer in 1970. So now it is called as Siemens. With each generation, so the instruments increased in size and capabilities. So in the early 1980s, a revolution began when the first fully automated laboratory emerged, and automation spread steadily throughout the 20th century. Advances in laboratory automation evolved and integrated into all aspects of laboratory medicine, from pre-analytical, which involves collection, receiving, and storage; analytical, the testing phase itself; and post-analytical, which involves archiving or reporting processes. So laboratory information systems and management systems enhance automation. Today, almost every laboratory area has some form of automation assimilated into its process. So, some of the events regarding the history of automation. So, way back 1976, it started the production of thin film analysis technology, and the first company that used microsample volumes and reagents on slides for dry chemistry analysis was Kodak Ektachem, which is now Vitros. And they were also the first to incorporate computer technology extensively into its design and use. Now let's proceed to the basic concepts and approaches in automation. So please take note of the different factors that serve to drive laboratory automation. So there are a lot. So please become familiarized with the different factors presented. There are many advantages to automating procedures. So one purpose is to increase the number of tests performed by one laboratorian in a given period. Labor is an extensive commodity in clinical laboratories. So through mechanization, the labor component devoted to any single test is minimized, and this effectively lowers the cost per test. A second purpose is to minimize the variation in results from one laboratorian to another. So by reproducing the components in a procedure as identically as possible, the coefficient of variation is also lowered and reproducibility is increased. So accuracy is then not dependent on the skill or workload of a particular operator on a particular day. This allows better comparison of results from day to day and week to week. Automation, however, cannot correct for deficiencies inherent in methodology. A third advantage is gained because automation eliminates the potential errors of manual analysis such as volumetric pipetting steps, calculation of results, and transcription of results. A fourth advantage accrues because instruments can use very small amounts of samples and reagents. This allows less blood to be drawn from each patient, and the use of small amounts of reagents decreases the cost of consumables. In addition, automation facilitates better space utilization through consolidation. So aside from those mentioned, automation also improves laboratory ordering, testing, reporting. This is accomplished through higher throughput, higher production rates, increased productivity, efficient use of reagents and materials, and standardization, which decreases the frequency of outliers with reduction in errors, resulting in improved turnaround times. So overall, automation reduces production costs with a positive impact on service to patients while creating a safer environment for the staff. So this figure just shows the difference between a laboratory without automation, partial automation, and of course total laboratory automation. Now we have some terms to define. So we have dwell time, minimum time from initial sampling to the production of a result. Next one is throughput, so the maximum number of test results that can be produced by an analyzer in a given time period, which is usually an hour. So we also have standard turnaround time. It is defined by most of the laboratories as the time interval between the specimens received in the laboratory to the time of reports dispatched with verification. So the clinical laboratories need a continuous high-quality customer service in order for them to keep their high-quality serving. So these requirements are becoming increasingly questionable because of cost apprehensions. So it is thus very difficult for the clinical laboratories to balance cost with the quality goals, patient safety, and demands. So that is why we have three automated system designs. We have total laboratory automation, modular integrated systems, and the standalone systems. So let's define first total laboratory automation. So when we talk about total laboratory automation, it is an automation system for the performance of highly repetitive tasks in the laboratory. It replaces human operators in the preparation and transport of specimens. So with robotic devices, so laboratory automation consolidates the control of multiple different analytical instruments to a smaller number of operators. So this way, the automation reduces the cost in laboratory testing. So as what we have presented on our screen. So benefits of laboratory automation include reduction of human errors in specimen handling. It also improves the overall process control and offers faster turnaround times from specimen collection to test result reporting. Additionally, most clinical laboratories, in order to face the falling reimbursements for medical services, face a decision about implementing total laboratory automation or portions of it to reduce operating costs. So many clinical laboratories worldwide are converting to total laboratory automation. So there are strong evidence that automation not only heightens profitability but also improves quality, timeliness, and lab flexibility. After that, a great percentage of hematology and clinical chemistry analyze their tests completely automatically. So we have this FTE or full-time equivalent. So when we talk about this term, so it is the hours worked by one employee in a full-time basis. But aside from those advantages that we have mentioned about total laboratory automation, so we have the following major limitations also. So the need for substantial financial investments and increased floor space, the need for highly technical personnel to operate and troubleshoot the system, personnel team building, and infrastructure remodeling. Next, we have one company that really is employing total laboratory automation. So we have the Roche Diagnostic Systems. So they have employed modular pre-analytics and platforms, so such as in our chemistry analyzers. So even though if you are planning to really have an initial investment with this kind of machines, so your money may reach millions of dollars, because this is US based, and the floor space requirements may exceed 4,000 square feet because Roche Diagnostic System or our chemistry analyzers are humongous, to fit inside the laboratory. Next, we have our modular integrated systems. So this one links together multiple laboratory disciplines into a single testing platform that is interconnected by a track. So when compared to our total laboratory automation, you are going to need less capital investment. Okay. Lastly, we have the standalone system. So in general, in quality control laboratory instruments attached with a personal computer without LAN or WAN connections are considered as standalone. So the standalone laboratory is a station part which has everything necessary to serve as one-piece functional space station. So therefore, to automate specific sections of the process which are still manual operations. So that's your standalone system in the laboratory. So we have different classifications of automated analyzers, but there are three basic approaches with instruments. So we have the continuous flow, we have the centrifugal analysis, the discrete analysis. So all of those three mentioned can use batch analysis, which involves large number of specimens in one run, but only discrete analyzers offer random access or stat capabilities. Now let's proceed with your continuous flow analyzer. So the samples are introduced in a sequential manner, following each other through the same network. So series of air bubbles at regular intervals serves as separating end media. So the internal diameter of the tubing and the rate of flow determine the volumes of sample prior to mixing the reagents and the turnaround time of the result. So continuous flow therefore resolves the major consideration of uniformity in the performance of tests because each sample follows the same reaction path. Continuous flow also assists the laboratory that needs to run many samples requiring the same procedure. So in continuous flow, liquids such as the reagents, diluents, and samples are pumped through a system of continuous tubing. So samples are introduced in a sequential manner, following each other through the same network. So as what I have mentioned earlier. So therefore, as I recall, from what I have mentioned earlier, a series of air bubbles at regular intervals will serve as a separating and cleaning media. Continuous flow therefore resolves the major consideration uniformity and performance of tests because each sample follows the same reaction path. So the major drawbacks that contributed to the eventual demise of traditional continuous flow analyzers in the marketplace were significant carryover problems and wasteful use of continuously flowing reagents. So Technicon's, which is now Siemens, answer to these problems was a non-continuous flow discrete analyzer, which was the RA1000, using random access fluid or a hydrofluorocarbon liquid to reduce surface tension between samples or reagents and their tubing, and thereby reducing carryover. So later, the Chem 1 was developed by Technicon to use Teflon tubing and Teflon oil, virtually eliminating carryover problems. So the Chem 1 was a continuous flow analyzer, but only remotely comparable to the original continuous flow principle. So this is how continuous flow analyzer works internally. Now let's proceed to the discrete analyzer. So discrete analysis is the separation of each sample and accompanying reagents in a separate container. Discrete analyzers have the capability of running multiple tests, one sample at a time, or multiple samples, one test at a time. So they are the most popular and versatile analyzers and have almost completely replaced continuous flow and centrifugal analyzers. However, because each sample is in a separate reaction container, uniformity of quality must be maintained in each cuvette so that a particular sample's quality is not affected by the particular space that it occupies. So the next one, we have our centrifugal analyzer. So centrifugal analysis uses the force generated by centrifugation to transfer and then contain liquids in a separate cuvette for measurement at the perimeter of a spinning rotor. So centrifugal analyzers are most capable of running multiple samples but one test at a time in a batch. So batch analysis is their major advantage because reactions in all cuvettes are read virtually simultaneously, taking no longer to run a full rotor of about 30 samples than it would take to run a few. Laboratories with a high workload of individual tests for routine batch analysis may use these instruments. Again, each cuvette must be uniformly matched to each other to maintain quality handling of each sample. So the Cobas Bio or the Roche Diagnostics with a xenon flash lamp and a longitudinal cuvettes and the IL Monarch with a fully integrated walkaway design are two of the more successful centrifugal analyzers. Now we also have sequential. So the classification of automated analyzers, so we have sequential. When we talk about sequential analysis, it means that all samples are processed in the order that they are received and multiple tests are performed in a certain order. Next, we have batch analysis. All samples are loaded at the same time and a single test is conducted on each sample. The next one is a parallel analysis. So more than one test is analyzed concurrently on a given clinical system. And lastly is random access analysis. So it means that the test reactions can be programmed to occur in a different order than the samples are placed on the instrument, and specific tests can be programmed for each sample. So this is helpful when many samples are placed on an analyzer and are processing, but an urgent test request must be added later. So this shows our automated analyzers. Now let's proceed to steps in automated analysis. In clinical chemistry, automation is the mechanization of the steps in a procedure. Manufacturers design their instruments to mimic manual techniques. So the major processes performed by an automated analyzer can be divided into specimen identification and preparation, chemical reaction and data collection, and analysis. So operations generally occur in the order listed from 1 to 8 as shown on your screen. However, there may be slight variations in the order, so some steps may be deleted or duplicated. So most analyzers have the capability to dilute the sample and repeat the testing process if the analyte concentration exceeds the linear range of the assay. Each step of automated analysis is explained in this section, so several different applications are also discussed. So several instruments have been chosen because they have components that represent either common features used in chemistry instrumentation or a unique method of automating a step in a procedure. So none of the representative instruments is completely described, but rather, the important components are described in the screen as examples. Preparation of the sample for analysis has been and remains a manual process in most laboratories. The clotting time, if using serum, centrifugation and the transferring of the sample to an analyzer cup, unless using primary tube sampling, so cost delay and expense in the testing process. One alternative to manual preparation is to automate this process by using robotics on front-end automation to handle the specimen through these steps and load the specimen onto the analyzer. Another option is to bypass the specimen preparation altogether by using using whole blood for analysis. For example, so the Abbott Vision company, robotics for specimen preparation has already become a reality in some clinical laboratories in the United States and other countries. So another approach is to use a plasma separator tube and perform primary tube sampling with heparin plasma. This eliminates the need for both to wait for the sample to clot and to aliquot the sample. So the sample must be properly identified and its location in the analyzer must be monitored throughout the test. So the simplest means of identifying a sample is by placing a manually labeled sample cup in a numbered analysis portion on the analyzer, in accordance with a manually prepared worksheet or a computer-generated load list. The most sophisticated approach that is commonly used today employs a barcode label affixed to the primary collection tube. This label contains patient demographics and also may include test requests. The barcode labeled tubes are then transferred to the loading zone of the analyzer, where the barcode is scanned and the information is stored in the computer's memory. The analyzer is then capable of monitoring all functions of identification, test orders and parameters, and sample position. So certain analyzers may take test requests downloaded from the laboratory information system, or the so-called LIS, and run them when the appropriate sample is identified and ready to be pipetted. So we also have emerging technologies. So we had the automated specimen inspection. So it identifies sample identification errors and sample integrity issues, and it could sort a random collection of different size containers with different additives. We also have radio frequency identification. So it is passive and without active human intervention. So the advantages are presented as follows: no line of sight reading, dynamic data storage not affected by cold storage. Disadvantage, so it costs higher, and then it is yet to be commercialized on a large scale in the clinical laboratory. So most instruments use either circular carousels or rectangular tracks as specimen containers for holding disposable sample cups, or primary sample tubes in the loading or pipetting zone of the analyzer. So these cups or tubes hold standards, controls, and patient specimens to be pipetted into the reaction chambers of the analyzer. So the slots in the trays or racks are usually numbered to aid in sample identification. So the trays or racks move automatically in one position steps at a pre-selected speed. So the speed determines the number of specimens to be analyzed per hour. As a convenience, the instrument can determine the slot number containing the last sample and terminate analysis after that sample. So the instrument's microprocessor holds the number of samples in memory and aspirates only in positions containing the samples. So next we have the electric track vehicles. So larger carrying capacity than pneumatic tube systems, and it is not associated with problems such as damage to specimens caused by acceleration or deceleration of forces. We also have our mobile robot. So delivery of systems to lab benches by a mobile robot is usually more frequent than human pickup and has been shown to be cost effective. That's our mobile robots. And this one is our electric track vehicles. It looks like a cable car. So it moves swiftly on the ceiling. And just like that, so that's how it works. Okay, sorry, so now to continue. So the barcode labeled tubes are then transferred to the loading zone of the analyzer, where the barcode is scanned and the information is stored in the computer's memory. So the analyzer is then capable of monitoring all functions of identification, test orders and parameters, and sample position. So alternatives of specimen preparation and identification are presented on your screen. So circular or carousels or rectangular tracks as specimen containers for holding disposable sample cups or primary sample tubes in the loading or pipetting zone of the analyzers can be used for specimen loading and aspiration. So I have already mentioned this one in the previous slide. So this is how it looks like, everyone. So for figure 7-2, this is the Vitros machine. So there are 4 quadrant trays. And then for figure 7-3, it is the Roche or the Hitachi 5-position rack. So sampling probes on instruments using specific sampling cups are programmed or adjusted to reach its prescribed depth in those cups to maximize the use of available sample. So for specimen processing, autoanalyzers should be capable of removing protein and other interferents. So such as in the following examples, we have dialysis, column chromatography, and filtration. Now let's proceed to the reagent system and delivery. open system, the operator has the advantage that he can purchase reagents from any company, so he can save money by buying reagents which are cheaper, thereby reducing the cost per test. Closed system, so in this system, the operator has to buy chemicals only from a particular company, because reagents from different companies will simply not be accepted by the machine. So machine will not run, so operator cannot reduce the cost per test. So next, reagents have to be provided in unique containers or format as prescribed by its manufacturer. So these two adds to cost per test. However, it allows high degree of automation. So reagents may be classified as liquid or dry systems for use with automated analyzers. Liquid reagents may be purchased in bulk volume containers or in unit dose packaging as a convenience for stat testing on some analyzers. Dry reagents are packaged in various forms. They may be bottled as lyophilized powder, which requires reconstitution with water or buffer. Unless the manufacturer provides the diluent, the water quality available in the laboratory is important. A second and unique type of dry reagents is the multi-layered dry chemistry slide. So for the Vitros analyzer, it was rebranded in 2001 as the Vitros microslide technology. So these slides have microscopically thin layers of dry reagents mounted on a plastic support, so the slides are approximately the size and thickness of a postage stamp. Reagent handling varies accordingly to instrument capabilities and methodologies. Many test procedures use sensitive, short-lived working reagents, so contemporary analyzers use a variety of techniques to preserve them. One technique is to keep all reagents refrigerated until the moment of need and then quickly pre-incubate them to reaction temperature or store them in a refrigerated compartment on the analyzer that feeds directly to the dispensing area. Another means of preservation is to provide reagents in a dried tablet form and reconstitute them when the test is to be run. So a third is to manufacture the reagent into stable components that will be combined at the moment of reaction. So if this approach is used, the first component also may be used as a diluent for the sample. So the various manufacturers often use combinations of these reagent handling techniques. So here are our examples of our reagents. So we have the dipstick. We also have the Fujifilm company for producing other reagents. So this is how a dry slide technology looks like. So here are the 3 or more layers of our dry slide technology. So the first one is the spreading layer. The second one is a central layer. And then the third one is the indicator layer. So the number of layers varies depending on the assay to be performed. So the reagent layer contains enzymes, dye precursors, and buffers necessary for the analysis of a specific component. The sample, control, or standard is deposited on the spreading layer. And then selected components are allowed to penetrate to the reaction layer, which in turn activate the dehydrated reagents. So we also have techniques of preservation. So you should keep all reagents refrigerated until the moment of need and then quickly pre-incubate them to reaction temperature or store them in a refrigerated compartment on the analyzer that feeds directly to the dispensing area. So I have already mentioned this one on the previous slide earlier. So the various manufacturers often use combinations of different reagent handling techniques. So there are here are the different delivery techniques. So we have syringes, we also have piston-driven pumps, we also have pressurized reagent bottles. So we also have the chemical reaction phase. It consists of mixing and separation. For mixing, it involves forceful dispensing, magnetic steering, vigorous lateral displacement, rotating paddle, and ultrasonic energy. While separation uses a very high reagent to sample ratio and shortens the reaction time to eliminate slower reacting interference. Regardless of the technique used, mixing is a difficult process to automate. Next, we have our incubation and reaction time. When we talk about incubation, it uses a heating bath, which will maintain the required temperature of the reaction mixture. And then the reaction time, on the other hand, it is the completion of a reaction or the rate at which the reaction is proceeding. So the measurement phase, it uses the following. We have the UV light, fluorescent, flame photometry, ion selective electrodes, gamma counters, and luminometers. So the most common methods for measurement phase are still visible and UV spectrophotometer. So the advantage of calibrating an automated instrument is the long-term stability of the standard curve, which requires only monitoring with controls on a daily basis. Okay? So steps in automated analysis. So this was already presented earlier. So just a recap from steps 1 to 8. And then this table, this box rather, shows the pre-analytic and post-analytic steps in automation. Now let's proceed to the future trends in automation. So automation will continue to evolve, and then system integration and miniaturization with more technologically advanced computer power will persist to accommodate more portable analyzers for more precise testing. And then automated analyzers will have artificial intelligence, whereby the computer will think or make decisions if sufficiently programmed with infinite scenarios of data. And then spectral mapping or multiple wavelength monitoring with high-resolution photometers and polychrometers will become standard. So in the near future, so hopefully all of this will become a possibility. And with that, that is the end of our discussion, everyone. So thank you so much for listening. So if you have questions or clarifications, please do not hesitate to message me. So thank you so much and have a good day.